I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before starting the topic, please like, share, and subscribe the topic. In the last session, we dealt with denaturation and precipitation reactions of proteins. In today's session, we will study about classification of proteins. Classification based on functions. Catalytic proteins, example enzymes. Structural proteins, example collagen and elastin. Contractile proteins, example myosin and actin. Transport proteins, example hemoglobin, myoglobin, albumin and transferrin. Regulatory proteins or hormones, example ECTH, insulin, growth hormone. Genetic proteins, example histones. Protective proteins, example immunoglobulins, interferons and clotting factors. Classification based on composition and solubility. In this, first is simple proteins. According to definition, they contain only amino acids. First, albumins. They are soluble in water and coagulated by heat. Human serum albumin has a molecular weight of 69,000. Other examples are lactalbumin of milk and egg albumin. Second is globulins. These are insoluble in water but soluble in salt solutions. They are also coagulated by heat. Examples are egg globulin, serum globulins, and legumin of peas. Protamins. These are soluble in water, dilute alkalis, and acids. They are not coagulated by heating. They contain large number of arginine and lysine residues and so are strongly basic. Hence, they can combine with other acidic proteins. Protamine zinc insulinate is a common commercial preparation of insulin. Fourth one is prolamins. They are about soluble in 70 to 80% of alcohol but insoluble in pure water. They are rich in proline but lack in lysine. Examples are zein from corn, gliadin of wheat, and hordine of barley. Next is lectins. They are precipitated by 30 to 60% ammonium sulfate. They are proteins having high affinity to sugar groups. Example, lectin from Dolichos biflorus which will agglutinate human blood group A1 RBCs. Phytohemagglutinin or PHA, a lectin from Phaseolus vulgaris, that is red kidney bean, agglutinates all RBCs and WBCs. Concanaverin or corn A from legumes will specifically attach to mannose and glucose. Then there is scleroproteins. They are insoluble in water, salt solutions and organic solvents, but soluble only in hot strong acids. Example, collagen of bone, cartilage and tendon, and keratin of hair, horn, nail and hoof. Conjugated proteins. They are combinations of proteins with a non-protein part called prosthetic group. Glycoproteins. They are proteins combined with carbohydrates. Hydroxyl hydroxyl group of serine or threonine and amide groups of asparagines and glutamine form the linkages with carbohydrate residues. Next is blood group antigens and many serum proteins are glycoproteins. When the carbohydrate content is more than 10% of the molecule, the viscosity is correspondingly increased, and they are sometimes known as mucoproteins or proteoglycans. The second example is lipoproteins. These are proteins loosely combined with the lipid components. They, they occur in blood and on cell membranes. Conjugated proteins. The third one is nucleoproteins. They are proteins attached to nucleic acids. Example, histones. The DNA contains negative charges which combines with positively charged proteins. The fourth one is chromoproteins. These are proteins with a colored prosthetic groups. Example, hemoglobin, which contains heme and gives red color. Flavoproteins, it contains riboflavin and gives yellow color. Visual purple, which contains vitamin A and gives purple color. Phosphoproteins, these contain phosphorus, example casein of milk and betaine of egg yolk. The phosphoric acid is esterified to the hydroxyl group of serine and threonine residues in proteins. Next is metalloproteins. They contain metal ions. Example, hemoglobin contains iron. Cytochromes also contains iron. Tyrosinase contains copper and carbonic anhydrase contains zinc. Examples of conjugated proteins. Hemoglobin contains globin as a prosthetic protein part and heme as having prosthetic group. Nucleoprotein contains histones as protein part and DNA as a prosthetic group. Rhodopsin contains opsin as protein part while 11-cis retinol as prosthetic group. Succinate dehydrogenase contains enzyme which is protein part and riboflavin which is prosthetic group. Ferritin which contains apoferritin as protein part and iron as prosthetic group. Ceruloplasmin contains apoceruloplasmin as protein part and copper as a prosthetic group. They are degradation products of native proteins. Progressive hydrolysis of protein contain smaller and smaller chain. Like protein is first broken down into peptones, then into peptides, and finally the amino acids. Classification based on shape of proteins. First, the globular proteins. They are spherical or oval in shape. They are easily soluble. Examples are albumins, globulins, and glutamins. Next one is fibrous proteins. The molecules are elongated or needle shaped. Their solubility is minimum. They resist digestion. Example, collagen, elastin, and keratins. Classification based on nutritional value. In that first is nutritionally rich proteins. They are also called as complete proteins or first class proteins. They contain all the essential amino acids in the required proportions. On supplying these proteins in the diet, children will grow satisfactorily. Examples, casein of milk. Incomplete proteins. They lack one essential amino acid. They cannot promote body growth in children but may be able to sustain the body weight in adults. Protein from the pulses are deficient in methionine while proteins of cereals lack in lysine. If both of them are combined in the diet, adequate growth may be obtained. The third one is poor proteins. They lack in many essential amino acids, and based on these proteins will not only sustain the original body weight. Example, zein from corn lacks tryptophan and lysine. Biologically important peptides. When 10 or less number of amino acids are joined together, it is called oligopeptide. Some of them are biologically active. A few examples are thyrotropin releasing hormone or TRH. It is a tripeptide with the sequence of glutamic acid, histidine and proline. But the glutamin glutamic acid is further modified to form pyroglutamic acid. Biologically so the glutathione. The next example is of glutathione. It is a tripeptide. It is gamma glutamyl cystinyl glycine. It is involved in erythrocyte membrane integrity and is important in keeping enzymes in active state. The third example is oxytocin and vasopressin or ADH are nanopeptides with 9 amino acids. They are secreted by posterior pituitary. Fourth is angiotensin 1 which has 10 amino acids and angiotensin 2 which has 8 amino acids. They cause hypertension. Fifth one is trimethylene C, an antibiotic produced by Bacillus brevis, which contains 10 amino acids in the sick. It is circular and contains D-phenylalanine. Here ends our session. Please like, share, and subscribe the channel. Thank you.